Hello, everyone, and welcome to the Arima webinar, Illuminating the Human Retina with 3D Genomics. So today we'll have two presentations. The first will be a short introduction to 3D genomics and where it fits in within other omics approaches. And that's going to set the stage for our guest speaker, Dr. Jimena Corso Diaz, who will share about her lab's recent work on leveraging chromatin architecture to understand blinding diseases. And then after that, we're going to wrap it up with a Q&A at the very end. So please keep those questions coming for a lively discussion afterwards. We'll get to all the questions at the very end of the presentations. Now, I'd like to introduce our first speaker and my colleague, Pamela Bentley Mills. Pamela leads content strategy and creation at Arima Genomics to educate and engage the scientific community on what we like to call the power of 3D genomics to drive discovery and human health. Pamela received her PhD from the University of Alberta, where she worked on elucidating the effects of photoperiod and circadian rhythms on mammary gland development, function, and transcriptional regulation. Pamela, the floor is yours.
Wonderful. Well, I'm so happy to be here to talk a little bit about how 3D genomics can strengthen human health and disease research in general, particularly today on February 28th, Rare Disease Day. So as you may know, there are over 6,000 rare diseases that have been identified, with about 72% of them having a genetic origin. And of those 6,000, about 200 are rare cancers. And I'll talk through a couple examples today of some rare cancers that have been investigated with 3D genomics. But please feel free to post on social media if you've been affected by rare diseases or if you participate in Rare Disease Day using the Rare Disease Day hashtag. For my talk, I'll give a brief introduction to Arima and 3D genomics in general. I'll share a bit about our technology and then spotlight these two rare cancers. For those of you who are new to the Arima community or 3D genomics, welcome. We're so glad to have you. Arima is a small company based in Carlsbad, California, and we provide kits and reagents as well as end-to-end services to help scientists address a variety of fundamental biological questions, everything from basic science through clinical research. So what is 3D genomics? Well, let's start out with what we're all familiar with, which is linear genomics. This is the process of sequencing the genome to figure out the order of each and every one of the 3 billion base pairs in, say, the human genome, ideally in one long straight line. But of course, there's actually far much more complexity to the genome than can be captured in this very simplified linear paradigm. And that's where 3D genomics is very useful. So we know that those 3 billion base pairs are actually stretched to about 6 feet long, and they're folded up and packed into this tiny 5-micron nucleus in every single cell in our body. But that DNA is not randomly contained in the nucleus of cells. There is a defined pattern for how chromosomes are packaged into compartments so that they can be readily transcribed or not. And 3D genomics takes into account not only the sequence, but the structural organization of DNA in the nucleus. And this really helps better understand the regulatory landscape to ultimately understand genome function. Taking a bit more of a closer look at 3D Genome Organization, we can see that chromosomes highlighted here by different colors are organized into territories. These are then sub-organized into compartments with an inactive B compartment and a more active A compartment. Those are then comprised of domains and specifically topologically associated domains or TADs. And then within the active TADs, there are these looping structures. And this is where most of the regulatory interactions take place and are mediated through the assistance of various architectural proteins and histones. So hopefully I've gotten you all excited about 3D genomics, but what can you actually do with 3D genomics data? Well, quite a bit. So everything from assembling chromosome scale genomes to understanding the basic function of how genomes work and evolve, interpreting disease risk variants, and Jimena is going to go through a really beautiful example of this in her work shortly. You can discover mechanisms of gene regulation, and you can also detect structural variants and clinically relevant gene fusions with Hi-C. So how does 3D genomics really fit into the multi-omics landscape that we all need to be thinking about these days? Well, really it sits between genomics, which is looking at just the sequence, and epigenomics, which are those non-sequence modifications to the genome. And as you will see today, I'm going to share several examples of how combining 3D genomic data with, say, RNA-seq data can really help elucidate what is happening in a disease state. And so one of the common ways that we see our customers using a multi-omics approach is by combining 2 or 3 of these technologies. So oftentimes people will have transcriptomic data. So they know that there is differential gene expression between, say, 2 disease states. They may follow that up with a TAC-seq or ChIP-seq to identify what regions of the genome are being differentially regulated via chromatin accessibility. And then they apply 3D genomics, which really helps them resolve the mechanisms of that long-range gene regulation. And so we see this as providing a really comprehensive understanding of the complexity of gene regulation. And this can be used across a variety of disease states, as we'll talk about today. So to share a little bit specifically about our technology, Arima technology is based in Hi-C, or high throughput chromatin conformation capture. At Arima, we offer a range of products that allow scientists to look at both a genome-wide scale or at specific regions of the genome, or assemble the genome to get a chromosome scale assembly. Our technology is essentially a sample prep methodology. So really, you can start with a wide variety of sample types, everything from blood, frozen tissue, and FFP samples. And what is being done with the sample prep kit is basically linking any pieces of chromosomes that are in proximity to each other in the nucleus together so that they're bound together. Then through standard library prep and NGS sequencing, we can get Hi-C data, which I'll show you a little bit of shortly. And through data analysis, we can pull apart what are the 3-dimensional structures that are happening in that nucleus, even though we were able to sequence it as if it was a linear piece of DNA. Some of the key advantages of our technology are that it plugs directly into existing NGS ecosystems and workflows, so you don't need to purchase additional lab equipment to do this. And also, our methods are very robust and reliable and provide really insightful results across a wide range of tissue types and disease states. Okay, so this is Hi-C data, and I believe Jimena will be showing you a couple heat maps as well. So I want to just give you an introduction to this data type. What you're seeing here is all of the chromosomes of the human genome lined up on the X and Y axis. And this is a normal genome that was sequenced as part of the complete human genome by the T2T consortium and published in Science last year. And what you're seeing here is red signal that indicates interaction data. So the color represents the number of reads that support a 3-dimensional linkage between 2 loci of the genome. And so what's clear here is that there is a lot of signal showing intra-chromosomal interactions. So between chromosome 2 and chromosome 2, there's a lot of interaction, but there is very little interchromosomal interaction. So between chromosome 2 and say chromosome 14 out here, we can see that there is very little signal, and therefore we can interpret that as there not being a lot of three-dimensional interaction. So now what I've done is zoomed into a very small section of chromosome 2. And what you can see is, although like you can actually get quite a bit more resolution than when you look at the whole genome all at once. And so if we overlay a green, purple, gray, and blue dot over this just to highlight regions of this locus, this is what we would be thinking about in a linear model. So we would see that the purple is a little bit farther away from the green than the green is to the gray and the blue. However, with high-C data, what we're really doing is looking at interactions between regions of the locus. And so what we're saying is, does this region interact with this region? And so by overlaying this square, you can see that there is substantial signal here indicating that there is a three-dimensional interaction occurring. And what that looks like diagrammatically is that there is a loop being formed between the purple and the green dot, whereas a closed loop is not formed between, say, the green and the gray dot. And so really this can, these are the features that I'll talk through a couple papers about, and I'm sure Jimena will as well, but this is the sort of the data type that we use to understand the 3D genome and therefore tease out that gene regulation, specifically in diseases. So there's quite a breadth of research that's being done today with 3D genomics. So everything from just that basic understanding of the organization and function of the genome. Jimena will talk through a really nice example of disease risk variant interpretation, and then exploring disease mechanisms. So whether looking to understand evolution of a disease over time, identify drivers of disease, look at microenvironments, or establish biomarkers. And this is just a small selection of publications that we find really exciting, but scientists are using this technology across a wide range of fields as well. So everything from stem cell biology to psychiatric disorders, of course, cancer, heart and cardiac disease models. And this is one of the papers that I'll actually go through next, but it's a T-cell acute leukemia is a rare disease that also happens to be a type of cancer. And if you'd like to find publications in your research area, you can visit arimagenomics.com/publications. So one study that really clearly demonstrates the utility of 3D genomics to understand disease mechanisms is this paper published by Cochin et al. In Nature Genetics in 2020. And they were seeking to understand the mechanisms of this rare form of cancer, T-cell acute lymphoblastic leukemia. So what they did is, first they looked at a healthy cell and they focused in on the MYC locus. We know that MYC is an oncogene, and in healthy people, it is about 2 megabases away from its enhancer. And in three-dimensional space, we can see that it organizes into 2 independent loops. And looking more closely at the high-C data for this locus, you can see that there's the MYC gene right here interacting primarily with itself in its own little loop. And then there is its enhancer, which is down here, again, mainly self-interacting and not interacting with MYC. However, when they looked at people with TALL, what they saw is transcription factor binding to this enhancer actually alters the 3D conformation of this region of the genome, bringing the enhancer into close proximity with the oncogene. And so what this looked like with Hi-C data is that now you can see this MYC location and the enhancer are actually connected in three-dimensional space. And so that's where you see this coalescence of the two loops into one. And they showed that this, using RNA-seq, they showed that this actually correlates very closely with MYC gene expression. The authors of the study then went on to show that by inhibiting this transcription factor binding here with pharmaceuticals, they were able to restore the healthy state of the looping structure. And so this example really highlights that they were not only able to identify the mechanisms by which MYC is misregulated in T-cell acute lymphoblastic leukemia, but also inform exactly what transcription factor needed to be targeted in order to restore normal function. And so this is a great example of how using long-range linkage information can provide insights into oncogene regulation and be used to predict potential therapeutic effects. The next example I'll share with you is from Ewing sarcoma. This is a rare cancer affecting the bones, primarily of children, and it's characterized by this sort of moth-eaten appearance of the bone on X-ray. And it is the second most common bone cancer, with about 200 children being diagnosed per year in the United States. We know that this disease is caused by a gene fusion between the FET gene products and the ETS transcription factors. The most common fusion protein that results from this is this EWS-FLI1, which I'll just call EWS because it's quite a mouthful. And so this is the most common fusion that accounts for about 80% of the disease, and we know that it dysregulates transcription, leading to oncogenesis. So what a team at Nationwide Children's Hospital sought to understand is how this EWF fusion protein alters the 3D chromatin landscape in EWS cells. They created a series of knockdown and rescue models and combined Arima Hi-C with ChIP-seq and RNA sequencing to really elucidate the disease mechanism of what was causing this oncogenic transcriptional state. So here's what we're looking at is compared with the last example I showed you, we were looking at a very small MYC locus. Now we're looking at a much larger portion of the genome. And so this is an EWS-FLI knockdown, and these are individual TADs. So we can see that there's TADs that are in an active state in the A compartment and in the inactive state in the B compartment, so they're more coiled up. There is no binding of this EWF factor. And so this is what these cells look like when there is no EWS present. Now, contrast that with the wild type of, say, a Ewing sarcoma cell. And I know this is a little bit busy, but really what they're showing is that there are these very large changes in the three-dimensional structure of how this genome is organized. So you can see here that this TAD has flipped positions between the active and less active compartment. This TAD has moved from being the pink one, from being fully inactive towards becoming more active, and the chromatin has been made more accessible. You can also see that this EWS factor in green is binding and creating both intra-TAD loops that didn't previously exist, as well as inter-TAD loops. And so these data really highlighted a very key role of this EWS fusion protein in mediating genome-wide changes in chromatin conformation. And they really supported the notion that this fusion transcription factor serves as a master regulator of three-dimensional reprogramming of chromatin in Ewing sarcoma. And so, this is just a figure pulled from their graphical abstract, which I thought was a great way to show how these large changes can occur. But really, this research, has enabled them to see on a more genome-wide scale how this fusion protein is altering the chromatin landscape, and therefore, I'm sure now they are working on trying to figure out how they can return to a non-Ewing sarcoma-like state, either with therapeutics or other methods. So with that, I'll conclude here, and I hope I've convinced you that there is so much more to the genome than just sequence, that 3D genomics is an effective part of your multi-omics toolkit and can be used to identify and elucidate disease mechanisms and interpret disease risk variants. And lastly, that 3D genome analysis enables a wide range of human disease states, including rare cancers. So with that, thank you so much.
Thank you, Pamela, for that great introduction to the 3D genome and for walking us through some of that research. Next, I am very excited to introduce our guest speaker for today, Dr. Jimena Corso Diaz. Jimena is a research fellow in the Swaroop Lab at the National Eye Institute. Her research aims to identify the mechanisms of gene regulation that maintain homeostasis in the mammalian retina. Jimena uses deep sequencing technologies, including Hi-C, to identify gene regulatory regions in molecules involved in retinal development, aging, and age-related diseases. Jimena, thank you for being here. The floor is yours.
Thank you for the introduction and the opportunity to present our work today. So in Swaroop Lab at the National Eye Institute, we are very interested in identifying gene regulatory regions that are involved in regulating retinal genes to understand the genetic control of diseases of the retina. So in this study, we generated contact maps of the human retina, and we use these maps to identify target genes of regulatory regions and to understand chromatin organization in the retina. So just first, I would like to share the tissue we work with. So the way we can see is thanks to photoreceptor cells that lie in the back of our eyes in this very thin neuroepithelia called the retina. So light first enters through the cornea and is focused by the lens and hits photoreceptor cells, which are comprised mainly of rods and cone cells. Rods allow us to see at low light levels, and cones allow us to see colors. So in the retina, photoreceptors transform light energy into electrical energy and transmit the signal to other neurons in the retina that further process this signal and then send that to ganglion cells that form the optic nerve that finally transmit light signals to the brain. And these retinal cells are very susceptible to environmental variation as well as to genetic variation. So here I just want to give you an example of two diseases where retinal cells degenerate. One is retinitis pigmentosa, which is the most common inherited retinal disease, and is progressive and affects approximately 1.5 million people worldwide. At least 80 genes have been found to be mutated in this disease. And it affects primarily broad photoreceptors. So people that have this disease first experience poor vision during the night, and then also loss of peripheral vision. When enough photoreceptors die, then they can also lose vision in the central retina, and this can lead to complete blindness. And another disease that is more common is age-related macular degeneration, and this is the leading cause of vision loss in people over 55 years old. And it is predicted that in 20 years from now, almost 300 million people in the world will have macular degeneration. So it affects the macula, which is a central part of our retinas. And in this, as this degenerates, we can also completely lose sight. It is a complex disease that involves multiple cell types, and also it is influenced by aging, genetics, and also environmental factors. But aging is the #1 risk factor for developing macular degeneration. So there is a strong genetic component that leads to retinal diseases. So there is a need to understand how retinal genes are regulated. And so we need to identify what parts of the genome act as regulators of gene transcription. And this is important because we still don't know how most of the retinal genes are regulated, what are the enhancers that come in close proximity to the promoter of these genes. This is important because more than 50% of inherited retinal degeneration cases have unknown etiology. So maybe it is possible that mutations in the enhancers of important retinal genes cause degeneration. Also, when we have identified genetic variants associated with retinal diseases such as AMD or glaucoma, most of these variants fall in the non-coding regions of the genome. So we don't know what are the target genes. So there is a need to understand these cis-regulatory elements in the retina. And the way we can identify them is by looking at chromatin signatures that are associated with active regions or enhancer regions. For example, accessible chromatin, histone modifications, and also direct contacts between these regions and coding regions of the genome. So to identify and map these regulatory elements in the retina to genes, we perform an integrative analysis using genetic and epigenetic information. So in this study, we generated chromatin contact maps from the retina of 4 human donors. And we use the Arima tissue Hi-C kit with some modifications for the retina. And we integrated the loop information that we obtained from these contacts with epigenetic marks, including different histone modifications and signals from open chromatin. We also integrated this data with information of transcription factor binding. And we then identify cis-regulatory elements in the retina, and we integrated these elements with genetic variants that are associated with diseases such as AMD and glaucoma. And we also integrated this with information that we have from previous studies that associate genetic variation to variable gene expression. First we wanted to see if the chromatin maps that we obtained in this study are specific for the retina. So we observed that there was higher correlation among our retinal samples compared to other cell types, including colon cancer cells, lymphoblastoid cells, and neurons from the anterior cingulate cortex. And we also observed that retina-enriched genes localized to the A compartment in the retina, which is the active compartment, but localized to the B compartment or the inactive compartment in neurons and cancer cells. So this suggests that our maps are specific for the retina. We were also interested in understanding if these maps are conserved. So there were published data sets for contact maps in mice. So we compare interactions between gene pairs that we observed or that were published from the mouse retina. And we observed that over 35% of these gene-gene interactions were also seen in the human retina. So an example of this is shown here on the right. In this synthetic region, which is a highly conserved region among human and mouse, we can see that EGF gene interacts with this EMP-EP gene in the human and also in the mouse. But we also observe that whatever we are seeing, or this low number of genes that are present in the mouse retina, are probably due to a low resolution of this data set. So we think that the conservation that we observe here is probably an underestimation due to the differences in resolution of the data sets. And we see here that when we look at topological associated domains, which are regions that are self-interacting, we can observe that the same regions are seen in the mouse dataset. However, it was not called as topological associated domains in this dataset, probably due to lower resolution. But we think that this data suggests that there is a high conservation in chromatin contacts between mouse and human retinal cells. We wanted them to identify cis-regulatory elements in our data set, since the chromatin contacts are probably bringing together regulatory elements to promoter of genes. So to identify cis-regulatory elements, we look for chromatin states using different histone marks and open chromatin signals. So we observe that promoter regions are high in open chromatin signals, as well as in histone marks associated with active chromatin. So we call this state a promoter, and then we call other regions that have variable active histone marks and open chromatin regions as enhancers, and then states that have high trimethylation of lysine 9 associated with repression, we call that as heterochromatin. And indeed, our promoter regions were enriched for transcription start sites, and promoter and enhancer regions were also enriched for retina-specific transcription factors. So this suggests that our calling for regulatory elements was accurate. And then we wanted to also identify super enhancers, which are regions longer than normal enhancers that have higher occupancy of cell type specific transcription factors, but are also characterized by higher levels of histone acetylation. So we identify over 1,000 super enhancers in the human retina, ranging from 10 to 300 kilobases. And we observe that these super enhancers overlap with over 50% of retina enriched genes and over 70% of rod enriched genes, suggesting that these super enhancers are specific for the retina, and this is in agreement with the role of super enhancers in regulating tissue-specific genes. So to further characterize these super enhancers, we look for binding of retina-specific transcription factors. And we observe that if we look at footprints, which are regions within open chromatin that are predicted to be bound by DNA binding proteins, we found that these accessible footprints with the super enhancers are indeed enriched for binding of retinal transcription factors, such as CREs and NRL. And interestingly, we also observed that super enhancers overlap with retina-specific transcription factors. So in this example, NRL is our main transcription factor in the retina that regulates the development of rod photoreceptors. And we observed that there is a super enhancer that overlaps with NRL and that binds many transcription factors that are known to regulate retinal development. So this forms a self-regulatory network where not only super enhancers are bound by cell type or tissue-specific factors, but are also overlapping with these factors, with these specific transcription factors. And this may be important for the establishment of cell type specific programs, as has been shown in other cell types. We later wanted to identify candidate transcription factors that can act in concert with retinal specific factors. In this case, NRL and CREs. So we actually identify different factors, candidate transcription factors that regulate super enhancers by taking footprints located in super enhancers that also contain NRL and CREs binding, and then performing motif discovery analysis. In this study, we found that indeed some transcription factors that are known to regulate retinal development along with NRL and CREs are identified, like estrogen receptor beta, but we also find some novel transcription factors that have not been studied in the retina, such as this zinc finger protein CBTB18, which has been shown to be important for brain development, but perhaps it's also important for retinal development, and it will be interesting to study. So we then wanted to characterize the super enhancers that regulate probably retina-specific genes in relationship with chromatin features, particularly with topological associated domains. We find, interestingly, that topological associated domains that do not contain super enhancers are more insulated compared to topological associated domains that contain super enhancers, as seen here. But Interestingly, if the super enhancer is at the edge of the dot, these dots are even less insulated as compared to topological associated domains where the super enhancer is inside the dot. So this suggests that super enhancers indeed influence the interaction of these domains with faraway regions. And intriguingly, we also find that genes associated with super enhancers that are in the interior of dots are related to retina-specific functions like phototransduction. Super enhancers that are located at the edge of the dot are related to stress response pathways. So we think that perhaps these super enhancers at the edge of the dot are more dynamic and need to respond quickly to different signals and are able to engage different regions in the genome and far away genes, whereas super enhancers that regulate retina-specific functions are more isolated and stable. We also wanted to characterize regions of the genome that have shown variation associated with different gene expression. So in a previous study in the Swaroop Lab, it was characterized these eQTLs, which are expression quantitative loci. So these eQTLs are polymorphisms that associate with variable gene expression. And in the retina, it was identified that around 14,000 single nucleotide polymorphisms are associated with changes in expression in 10,000 genes. And out of these 14,000, 4,000 were specific for the retina. So we wanted to further characterize whether these regions of the genome where these eQTLs are located are directly regulating these genes. So we first observed that most of the eQTLs that were identified in the retina are present in the active compartment or the A compartment. And we further characterize the eQTLs in respect to how far are they from the genes, how far are they from promoters, and whether they overlap with regulatory regions that we identify, or whether they make physical contacts with promoter of genes. So we found that over 2,000 eQTLs overlap with chromatin loops, and over half of them make contacts with promoter of genes, and 100 of them make contact with the gene that is predicted to be regulated by these eQTLs. And an example of this is this SNP in the PCK2 locus that forms a long-range interaction with PCK2. So this suggests that the relationship between this genetic variation far away from PCK2 and the expression of PCK2 is actually direct. We also were interested in looking at whether genetic variation that has been associated with AMD and glaucoma, it is actually directly regulating retinal genes. And we wanted to basically identify the target genes of variants that have been associated with AMD and glaucoma. So to perform this study, we took variants that were associated with AMD from genome-wide association studies. So that includes 52 lead variants and linkage disequilibrium variants, which are variants that are in proximity to these lead variants. And we looked at whether these variants overlap with chromatin loops. And we found that over 100 of these variants overlap with loops. That includes 4 lead variants. So this is an example of a variant that has been associated with AMD in the CTRB1 locus. And this variant, as you can see, is making contact with a faraway gene, CFDP1. So we propose that this variant can affect the expression of this gene, and this is the candidate gene that is involved in the development of AMD. In fact, CFDP1 gene is highly conserved in zebrafish. And in zebrafish, it has been shown that it regulates eye development. So this is a very intriguing candidate gene that warrants further studies in the context of AMD. Similarly, for glaucoma, we studied 127 lead variants and their linkage disequilibrium variants obtained from genomic-wide association studies. And we found that over 500 overlap with high C loops, including 12 lead variants. And this is an example of a variant in the PKHD1 locus that is making direct contact with TFAP2B gene. So it's possible that this variant is regulating this gene, and that this gene is the target gene that is involved in the development of glaucoma. And in fact, deletion of this gene in mice generates glaucoma-like phenotypes. So this type of integration allows us to identify target genes for retinal disease. In summary, we have generated a contact map of the human retina and integrated this with epigenetic signatures. And this is the first chromatin map that has been generated for human retina. And we hope that it will be a good resource for different laboratories to identify candidate CREs targets and also transcription factors that are involved in retinal development and disease. We use this map to understand properties of topological associated domains. For example, we found that superenhancers at the edges of TADs associate with low insulation and are enriched for stress response genes. We also use this map to identify physical links of eQTLs. And in fact, we found that 100 eQTLs regulate their candidate target genes. Also, we use this map to identify candidate genes for susceptibility variants linked to AMD and glaucoma. So finally, I would like to thank the NNRL Lab and Anu and Swaroop for giving us the opportunity to work in this project. And I would like to acknowledge the people involved in this study. Nivedita Sain is a biologist postdoctoral fellow that worked with the high C libraries, and Zach, Jayshree, and Claire contributed to bioinformatics analysis. Catherine Jagger was involved in the development of ATAC-seq libraries. Thank you for your attention, and I'll be happy to take questions.
With that, I would like to open the floor up to questions. For the Q&A, my colleague Allison Whitaker is also going to join us. Allison is our technical support lead and truly a high C expert. And I see we already have a few questions in the Q&A, so let's dive right in. The first question is from Golnar, and they're asking, what are the factors affecting the 3D interaction between regions of the locus? Anyone who wants to take it, Allison, or anyone else is welcome to.
Yeah, I mean, well, a lot of times when you have a TAD, some of the things that actually regulate the boundaries of a TAD are CTCF and the cohesin SMC, right? So those are the things, and we actually find that if you change some of like the orientation or take away some of the sites, it can actually change the TAD structures and locations of TADs.
Thank you. Next question from Dahong. Do you think these super enhancers are likely cell type specific? I think this is probably a question for Jimena. Do we have to do sorting and further experiments to figure out the specificity, or there may be some data integration that we can bypass this? It seems hard to understand an RGC disease by looking at retina high C and other omics, but sure there are substantial technical challenges.
Yes, so definitely we are working here with bulk tissue. So most of the super enhancers that we identify will be coming from rod cells that are actually the most abundant cell type in the retina. However, many of these interactions will also be present in many retinal cell types. So will be neuronal specific, and in order to really identify whether these super enhancers are really regulating those genes, we need to do more studies, and we are actually currently doing some of these studies to validate the regulatory regions that we identified. So you really need to go further, and this is just an indication that those super enhancers exist and that there could be, and what are the target genes, but more validation is needed, definitely, yes.
Wonderful, thank you. Another question for Jimena from Brendan. So given that the high C data are derived from a bulk retina sample, how biased is the contact map towards rod chromatin architecture? For example, what is the likelihood that a given TAD identified in your data is present exclusively in rods versus in all retinal cell classes? Great question.
Yeah, so this is similar to the previous question, I think. Definitely the rod photoreceptors are over 70% of retinal cell types. So most likely that the TADs that we are identifying represent these domains in rods. But we did identify association with other retinal cell types. So we think that we can still pick up these interactions from different cell types, but we definitely need to further validate. And I think that we are more confident of calling some interactions a rod-specific interaction than anything else, but there is still information in our data suggests interactions between other retinal specific genes. Yeah.
Thank you.
I think I would just add there, Janetta, that you can look at chromatin conformation on both a bulk or a single cell level. And so we have several customers who have developed methods for looking at chromatin conformation at a single cell level, and can point in the direction of papers if you'd like to do that.
Perfect. Thank you. I do see that we're right at the hour, so maybe let's do one or two questions. I know there's a lot. Thank you all for your participation, but I do want to be mindful of everyone's time here. So let's do two more questions. So another one for Jimena. Is there a distinction between merge looping data and TAD heat map data? I can see TAD regions linking to other TAD regions through the merge looping data, but I cannot see a larger triangle connecting the two TADs. Do you know which
So-
slide they're referring to? Oh, go ahead.
I'm not sure I understand entirely, but I think you're referring to these triangles. They are called as that by an algorithm, and there are different algorithms that call that. So when you see the drawing of a triangle, is when the TAT is called in the algorithm. So there has to be a certain number of interactions to be called a TAT. So even if you see a few interactions among two regions, it may not be enough to be called a TAT. Is that? I'm not sure if that is the question.
Okay, thank you. Let's do one more question. I see there was one question that's upvoted here, and maybe it's more of a general question for Allison. How many cells do we need for high C analysis?
So typically we need the number of cells that relate to about 500 nanograms to 5 micrograms of DNA. And we find that for most cell types, about somewhere between 2 to 3 million cells is enough for input, even as low as 1 million for standard input. We have had cases where people have gone as low as 50,000 cells and we're still able to call loops. But typically we like you to have at least 1 million or ideally like 2 to 3 million cells.
Okay, thank you for that. I know we're over time. There's still more questions. If we didn't get to your question, we'll try to follow up with you individually. Feel free to shoot us an email or we'll get back to you as well. That is all the time that we have for today. So thank you folks for tuning in, and shout out to all of you who stayed with us until the end of the hour. And as a reminder, we will email the recording of this presentation to you in the next couple days. Have a great rest of your Tuesday, everyone. Thank you.